Hello everyone, and a warm welcome to today's Focus On session provided by GenQA. We'll just give a couple of minutes for people to log on to the system, and in the meantime, just a few technical pieces of information. If you have a question or if you require clarification on the presentation, you can use the chat box on your website screen to submit a query. This might be useful as well if you have any technical difficulties. Today's presentation is our second webinar focused on ISCN, and I would like to introduce you to Dr. Ros Hastings, who has extensive experience in cytogenomics and in the use of ISCN. Ros is an author of the 2020 edition of ISCN, and she's now the chair of the ISCN Standing Committee. So I'll hand you over to Ros now. Thank you.
Thank you, Melody. There may be some problems with the connection. It just went fuzzy then. So if there are, if you can put it in the chat, and then I'll go back to the previous slide if that's needed. But hopefully we won't have any more technical problems. So yeah, I'm going to talk about ISCN principles. Thank you. So I'm going to explain where ISCN fits in relation to HGVS and HUGO. Some basic ISCN principles, so they're the building blocks on which you can work out most of the principles. It is ideal if you read the book in its entirety. And then we'll go into specific ISCN for karyotypes for fish, for RSA, regional specific assays, arrays, NGS. There then will be a little bit about the online version and the forum that's available in ISCN 2020, plus a quiz just to keep you interested. And then, of course, questions and answers at the end. So the Standing Committee look at all the comments that come from the community and assess whether or not they should be included in the new version of the ISCN. Jean McGowan-Jordan used to be the chair, and I've since taken over that responsibility. And I have a co-chair, which is Sarah Moore, and she will deputise for me when need be. We also have Joanne Dunnan from the HGBS committee on the Standing Committee for ISCN. This year, for the first time, there is an online version available. And if you buy a hard copy of the ISCN 2020, you get access to the online version for 12 months for free. And there's also a forum there, which I'll discuss later. From a participant's point of perspective, we will expect the use of ISCN 2020 this year. So the ISCN committee, which is here, obviously you feed in from the genetics community and we obviously feed back. Likewise for the HGVS, there's also a gene nomenclature committee, and there may be in the future an HGVS pathogenicity committee too. Now, all 4 groups will report to a newly formed HUGO Standards Committee, which has 2 representatives from HGVS. It has myself and the deputy from ISCN and representatives from HGNC, and in future from the HGVS Pathogenicity Committee. And the HUGO Standards Committee does report to the HUGO board. So it's a bit more joined up. So hopefully what will happen in the future is we won't get one group making decisions that affect the other. And that certainly happened recently in the fusion genes, and now there's going to be a new publication coming out shortly in Nature Genetics. So the principles are that if you have a karyotype, you'd have a metaphase, and you'd give the karyotype, which would be 46XY. This is a karyogram, and this is the ideogram. And the karyotype is the nomenclature that's used to describe normal or abnormal, constitutional or acquired chromosomal complement of an individual tissue or cell line. It usually refers to the chromosome level. When you look at the ideograms, they're obviously placed in order from 1 to 22, and then the sex chromosomes are next. And when we talk about these chromosomes, then we talk about the centromere, And then we talk about the P arm, which is the short arm, and the Q arm, which is the long arm, and these obviously are the telomeres. And in here, you have different regions and then different bands. And that's how we decide whether or not a breakpoint happens in a particular region or a particular band. Let's say you have the short arm, you have the long arm, your centromere. Punctuation, I'm going to start with this first of all, is there to help people understand what we're referring to, and it's consistent across the board. So a full stop is used, excuse me, to separate techniques. A comma is used to separate chromosome number, the sex chromosome constitution, chromosome abnormalities, excuse me, probes, all within nucleotide numbers to indicate thousands and millions. The semicolon is used in the short form to show a break and join with another chromosome, and a double colon is used to show a break and join within or another with another chromosome in the long form. The plus sign is used to show gain, and for fish, it shows that the locus is present on a specific chromosome, and the minus sign shows loss. The underscore is used to show loss or gain that's encompassed between the listed nucleotides, and that's used in arrays. The question mark is used where you're uncertain about the chromosome or the region, and I'll explain a bit more where you use the question mark later. The tilde is used for intervals or boundaries or a range, and you will often see that when there is some uncertainty too. And the forward slash separates clones and cell lines. Obviously, we're talking about constitutional cytogenetics, so we won't be talking about clones today. So we have quite a few terms, and if you go to pages 34 to 36 of ISCN 2020, you'll find a load of terms. There are about 3 pages of them. I'm only going to talk about the ones that most commonly we use, apart from obviously the ones we talk about where we're talking about a type of rearrangement. So MAT still means maternally inherited, PAT still means paternally inherited, and you can use INH when it's inherited, but you don't know from which parent. We have a new term, which is DMAT and DPAT, which is an unbalanced derivative from a maternal rearrangement or an unbalanced derivative from a paternal rearrangement. DN stays the same as de novo. You've got mosaic, chimera terms, and C for a constitutional abnormality in an acquired disease, but we won't be discussing that today. So the principles. Well, for normal results where you've got chromosomes, you'd expect to see 46 chromosomes, 23 pairs, 22 autosomes from chromosomes 1 to 22, and 2 sex chromosomes if you're female XX, and if you're male, XY. ISCN covers both whole genome and region-specific nomenclature, and so it has to vary according to the technique to a certain extent. So for normal results, when you've got a carrier type, the chromosome number is always given first, followed by the sex chromosomes. And what's changed for ISCN 2020 is that for all techniques now, the sex chromosomes are listed first. For array, it would be for a female array X, 1 to 22, close the brackets, times 2. But for a male, because you've only got 1 copy of each, you have to take that out of the brackets because it's not the same as the autosomes where there's 2 copies. And so you have the X and Y in brackets times 1, a comma, and then the autosomes 1 to 22 in brackets times 2. For the region-specific ISCN, you're only talking about a specific region, and so it's slightly less complicated in some ways. So for metaphase fish, you would put ish, give the chromosome region, and then put in brackets the probe times 2 if it was normal. Or if you've got multiple probes, you would put in brackets probe A, comma, probe B, comma, etc., and brackets times 2. For the interphase fish, you use the term nukish, but you will notice there is no space, and you immediately put the bracket after the ish. and you put the probe in ×2, or you can have probe A, comma, probe B, and the ×2 goes outside the brackets this time. So it's only when you've got a single probe that you keep it within the bracket for normals, and if it's got multiple probes that are normal, you put the ×2 outside the bracket. Obviously, if you're dealing with X chromosomes and Y chromosomes, it'd be ×1, unless it's the PAR region. So for the assays, such as the regional specific assays, you'd have RSA, open brackets, the regions tested, close brackets, ×2. And you will notice that in this one, and this one, there is no spacing, whereas in NUCISH, whereas in ISH, there is a space before the chromosome region. And that is because when you've got a bracket, you can put it right next to the technique. I will explain that in a bit longer, where you put the spaces. So for abnormal results, you first of all give the total number of chromosomes, then you give the sex chromosome constitution, and then thirdly, you give the aberrations. You use plus and minus to indicate gain or loss, and you use abbreviations for the type of structural aberrations. And the only exception to this is constitutional sex chromosome aberrations, which we'll explain later. For numerical changes, you do it in the chromosome number order. So X goes before Y, and then the autosomes 1 goes before 2, goes before 3, etc. to 22. So in these particular examples, you've got 47 chromosomes, comma, that's your sex chromosome constitution, that's female, comma, and you've got an additional chromosome 21, which makes sense. You've got 46 chromosomes, you've got an additional one, so it makes it 47. In this situation, with the sex chromosomes, you don't put a plus in. You actually give the sex as it is. And that's because in hematological, you would actually sometimes get gain or loss. But you're talking here about someone's actual sex complement, and that actually goes as it is. So if you've got pentasome X, it would be 50 XXXXX. Counted 5 there? Can't remember. Anyway, say 48 here. You've got an additional X chromosome, comma, plus 18, making it 48 chromosomes. But you do not put plus there. Here's another principle. You always describe numerical, then structural aberrations in the chromosome order. So if it's the same chromosome, you would put the numerical change before the structural change. So a plus 21 would go before a inversion 21. Otherwise, it goes in chromosomal order. So in this particular case, you've got 48 XXY, you've got an inversion 13, because you've got a normal 13, and this is the change of a chromosome 13. It's not plus, it's not additional, but you have got 3 copies of 21, so you've got plus 21. But also remember, with the sex chromosomes, you've got 48 chromosomes again, because you've got an additional 21. You've got the common inversion Y. You put a comma after the XX and then put inversion Y, but you don't put plus, because this is the sex chromosome constitution of the individuals. Because what's gained is actually the X, and then you'd have to put 48X plus X inversion Y, which doesn't make sense, and it sounds like it's an acquired abnormality. In fact, this is constitutional, and so that's why you don't have a plus in it. So you describe the abnormalities from Peta to Qta. So in this particular example, you've got 46X, Y with commas in between, inversion 7 from P13 to Q22. When you've got multiple structural aberrations of homologous chromosomes listed, you put these in alphabetical order. So here we go, numerical before structural. You have got 3 chromosome 8s in this particular patient, just so it makes sense to you. So you've got 47, comma, XY in a male, comma, you've got an additional 8, numerical before structural, comma, then you've got a deletion of 1 of the 8s, and then you've got an inversion in the other 8s. So you're separating each by a comma, and you put the structural aberrations in alphabetical order, so del goes before inversion. And in this particular example, I have put a rearrangement involving the sex chromosome. And because it's abnormal, it goes after the normal Y. So you've got 47Y, TX13. Then you've got your inversion 8, and then it's plus, because this is an additional chromosome, a DER13 from the TX13. And you don't have to repeat the breakpoints because it's the same rearrangement as described here. You can, but you don't have to. You can use both the short and detailed form. If you've got a complex carrier type, then you can interchange the two. The short form is far easier to use, but sometimes for clarity you do need the long detailed form. So this is an inversion of chromosome 7, but you could obviously put it from Peter through to Q22 to P13 to Q2, because that's what you're seeing. From Peter it's joining at Q22, and then you've got P13 and then to Q2. One of the changes that we have actually specified in ISCN 2020 is not to describe polymorphisms in ISCN. As we are going on to more technologies and we're finding more and more normal variants, it seems inappropriate to be describing normal variations in the ISCN, because from a clinician's point of view and from a patient's point of view, they will perceive this as being an abnormality when in fact it's normal variation. So other principles in structural abnormals. Aberrations without centromeres are described last, are not included in the chromosome number. So in this particular case, you've got 49 chromosomes because all of these have got centromeres. So you've got an inversion 13. Rings go before markers. Even though it's not alphabetical, they always go before markers because you don't know what these are and you can identify these. But this is the order that is defined in the ISCN book, because in this particular case, you haven't identified any of them. You use a slant line to separate different clones or subclones or cell lines. Cell numbers are only given for constitutional if they're mosaic, and you list them in the order of the size order. Normal cells are always listed last, irrespective of size. So I've given you an example here where the largest cell line is the normal cell line, but you have to list it last. The next largest, which is listed next, and there isn't a space there, even though it looks like there is 47, XXX, 10 cells, / 45X, 5 cells. The number of cells given in square brackets for mosaics. So in this particular case, it's another common rearrangement in constitutional 1122. You've got 15 cells there, and you give the normal cell line last. So if you've got multiple aberrations, how do you write them? And there's various ways of doing it. So in this particular case, if you've got a triploid cell line, which would normally be 69XXY or XXX or XYY, so you do have to give the sex chromosome so you know what sex you're talking about. That's all you put. But in this case, you've got a triploid with 5 copies of chromosome 22, so you just talk about the extra 2. So it's 71XXY plus 21 plus 21. If you have multiple copies of the same chromosome, structural, there won't be many examples in this in constitution unless you've got somebody who's consanguineous or you've got markers. And so you can, if you've got a consanguineous parents, write the translocation in the child where it's got 1 inherited from the father, 1 inherited from the mother, as ×2. Alternatively, you could separate them out and say MAT, write T25, Q21, Q31, MAT, comma, and put the other 225 and put PAT. Entirely up to yourselves. If you have a marker, it's 47XX plus marker because it has a centromere. If the markers are actually identical and you've got 2 of them, then you put 2 mar. If, however, they're different, you put plus mar1 plus mar2. And just to complicate things, if you had 3 markers, 2 of which were identical and 1 wasn't, you'd put 49XX plus mar1 times 2, because there's 2 of them, plus mar2. Actually, that should be 2 mar. Oh no, it's because mar1. Sorry, that's right. If you know the identity of the marker, then you can put it in brackets after MAR. So MAR 8 and MAR 18, and then the normal cell line last, even though it's the largest. Question marks. A question mark denotes uncertainty, and everything following it is unclear. So as I mentioned earlier, that's your P arm, this is your Q arm, here's your centromere. These are the regions 1, 2, 3, and then these are the bands. So if you have got, I've tried to do this as it's progressing through where the uncertainty is. If you're not sure that you've actually got an additional 21, then you'd put plus query 21. You think it's 21, but you're not sure. If, however, you think you've got a deletion 1, but you're not at all sure it's a deletion, and you're certainly not sure of the bands, then you put query beforehand. If you put del1 query, it means you know the breaks in the long arm, but the region and band are unsure. If you put del1 q query 2, you're saying there's a deletion in the long arm, but probably it's in the region 1q2, so it's in this region here. If you put query 2, 3, you're still uncertain if the break is in, where the break is in the region of 1q, but if it is in region 2, then it's in band 2, 3. In this particular case, you know there's a deletion in region 2, but the band within the region is unknown. You put query there. If you think you know what the band is, of course, you could put it in like this one and put it in as 3, because you know the breakpoint's in region 2, but you don't know about the uncertainty, whether it is actually region 3. So the question note goes before the region of uncertainty. Spaces are used in ISCN, but rarely. You do include a space after using MAT, PAT, DMAT, DPAT, in INH, DN, mosaic, chimera, heterozygous and homozygous. There are some interesting examples where things are slightly different. So when you're doing sex determination, whereas normally you'd put the chromosome in brackets, you do not for sex determination. And there are quite a few examples in the book where you'll see this. So this is an issue of a space here for chimera, then you put another space, then you have X, you put the probe times 2, and then the number of cells, slash, and then you have to put the X and the Y if you're doing it for sex chromosome determination. You put both of them in. You include a space after the technique, except when it's followed by the brackets, the sort of square brackets or the normal brackets. So you'd put ish bracket space, sorry, de 17, but you would have nuke ish and then bracket de 17 Z1 times 2. And there's no space between array and the genome build, but there is a space between the genome build and the X chromosome. However, you could put array, no space, 16q times 3, and there's no genome build for aneuploidies, for whole aneuploidies. For the RSA, again, no space. You put your X first, then your Y, and then 1318 times 21. And just, although we're talking about constitutional, you do include a space before and after con and sep if you're using those fusion gene probes. So when is a gain or loss included in ISCN when you're actually doing your analysis? We tend to use the hematological roles on clones. So 2 or more cells with the same gain or aberration can be reported. The exception is when a single cell can be reported when the same cell line was found in a previous initial or follow-up study of the same patient. And for a loss, because you can obviously get broken cells, it's 3 or more cells with the same loss. And the exception is when you've got 2 cells with identical losses of 1 or more chromosomes, when there's also present the same gain or structural aberration, then it can be considered clonal. So if we had the the translocation 1122 in most cells, but you've got a loss of the X chromosome because it's with a structural aberration, you could report that. The deletions. There is no semicolon used for deletions, duplications, or inversions within a chromosome in the short form. As I already mentioned for mosaics, I keep saying this because it's quite a common error that is made, that the 46XX cell line always goes last, even if it's the largest, and the largest abnormal cell line goes first. Some people like to abbreviate, but unfortunately ISCN doesn't allow these abbreviations. So whatever the deletion is, you can't put 5q minus or del 5q in the ISCN. You can use it in the text, but you can't use it in ISCN. Translocations. Well, there are different types of translocations. You can have reciprocal translocation, which is shown here, a balanced one. You can have whole arm ones where the break is actually at the centromere. You can have Robertsonian, where the satellite material of the acrocentric chromosome is lost, but that is inert and doesn't have any clinical effect if it is lost. And you can have insertional translocations, which involves an additional break. So with reciprocal translocations and with the others, you start with the lowest chromosome number first, and then you use a semicolon between the different chromosomes. So in this particular case, you start with chromosome 4, it's gone on to chromosome 8, and of course it's a reciprocal back from 8 to 4, and it's in 4p16, and the breakpoints are, and 8p23, and as I say, there's a semicolon between both. If you've got a more complex karyotype, which does happen in constitutional, and it's a translocation that involves more than 2 chromosomes, you start with the lowest number, X and Y, of course, before autosomes, and then 1 before 2, etc. And the order of the chromosomes is dependent on the movement of the chromosome segments. It's not something that you choose. So in this particular example, we start with the lowest. There's 4 chromosomes involved, 2, 3, 12, and 6. You start with 2. So 2q has gone on to 6q. So 2q has gone on to 6. 6q has gone on to 12. And 12q has gone on to 3. You do not put 2, 3, 6, 12, because otherwise you'll have no idea what the abnormality looks like. You have to follow the movement, and you always start with the lowest number first. Let's look at some of the fish ISCN principles. This often causes confusion. Hopefully we've made it a bit easier in the 2020 booklet. You do need to list the probes in numerical order from Peter to Cuta, and it's important that you check where the probes reside. The control probe is not given in ISCN if there's a normal signal pattern. The only exception being for sex determination, because you do need to know what the X and the Y signal pattern is. Normal FISH results can be reported in one string only if the same number of interphase cells was analysed from the different hybridizations. So if you're doing, let's say, an aneuploidy, and you've got a normal result, then you can list the X, Y, 13, 21, and 18 probes in one string. And you don't have to report them as different hybridizations. However, for abnormal interfaces, the fusion probes follow the chromosome order of the probe sets. So you have to keep them within the probe sets. So, same probe set. So the X and the Y and the 18 probe set is given before the 13 and the 21. Yes, 18 is after 13 and 21, but it's part of this probe set. So this is given first, followed by RB1 and the probe set for chromosome 21. Now this is a contig, and so it involves 3 different probes, and you put a slash or backslash between each of the probes. If you don't give it in the ISCN, it has to be given in the text, and that is actually written now in the ISCN 2020. For fish, the numerator, which is this, is the total number of abnormal cells, and the denominator, which is this, is the total number of cells analysed. So that includes the normals and the abnormals, and by default, you can work out how many cells were normal. When it comes to reporting fish, ×2 for both interphase and metafish ISCN. Metaphase fish includes the probe location, and it's optional for interphase fish. There are examples in the book. For sex determination, you have to include both probes. So you would have to put ISH-X and say there was no SRY present. There are examples where there's an abnormal X, a Y in the ISCN, so you do look at them all because they're not, there is a rule there as well. Abnormals, you put, if for the metaphases, plus or minus, or if you've got 2 extra signals, plus plus, or 3, plus plus plus. Structurally abnormal metaphase fish, you only describe the derivative chromosome. But if it's a mosaic, you describe both the abnormal clones. So you don't describe the normal if you've only got one abnormality, but if there's no normal, you have to describe both abnormal clones. And for interphase fish, you'd put ×3 or ×1, etc., not plus or minus. The difference is you can see the signal on metaphases, you can only count them on interphases. So you can see exactly where they are. So when do you change the breakpoints? You can change the breakpoints when on G banding, when fish or array extend the breakpoints from the original G banding. It's optional whether or not you repeat the breakpoints in the fish ISCN if they're the same as the G banded ISCN. These are all the same karyotype, just done slightly differently. The probe extends the breakpoint from P16 to P16.3. You could choose not to change the breakpoints here, or you could change it, or you could change it in both. And in this case, we haven't repeated the breakpoints because you don't need to. So all 3 are acceptable options. So when to use DMAT and DPAT? If you know the origin of the rearrangement, let's assume the child has got the unbalanced karyotype here, just to make it clear, and the mother has a balanced T411. So if you know mother has a balanced rearrangement and the child's got the unbalanced, you put DMAT before the fish. If it's cryptic, then the DMAT goes after the fish because that's how you identify it. If the mother also had the same unbalanced D4, then it wouldn't be DMAT, it would be MAT because they've inherited the same rearrangement. Let's look at the array and the regional specific assay ISCN. As I've already mentioned, you can combine short and detailed form of ISCN. When multiple techniques are used, each technique is separated by a full stop. And the order is as follows. G-banded goes first, followed by metaphase fish, followed by interphase fish, nukish, followed by array, followed by RSA, then sequencing. Now, obviously, for most cases, you're not going to use all those different techniques, but that is the order in which they should do. You should write them. So if you did G-banding and array, it'd be G-banded followed by array. If you did fish, array, and sequencing, it would be, let's assume it's metaphase fish, ish, array, sequencing. You can list the results using different techniques on separate lines, or you can tabulate them if they're complex results. But if it's a simple result, you are supposed to put it in one string. Now, I've shown you loads of results with mosaics where you put the number of cells you've analysed in brackets, which works for fish and works for G-banded. It does not work for array and RSA because you haven't got a number as such. You might be able to quantify the level of mosaicism, say it was 60%, in which case you would put it in brackets 0.6, but if you can't quantify it, you put 2 tilde to 3. So it's between 2 to 3 copies. You can use either option, it's entirely up to you. For regional specific assays, for abnormal results other than aneuploidy, if the nucleotide positions are available, then they should be included. However, if they're not, which many kits don't give you, you have to give the kit name and the version. And nucleotides can now be listed with commas again. It just makes it easier to work out what the numbers are at first sight. For the region-specific ISCN, again, because you've got brackets, it's RSA, bracket, X13, 18, 21, and bracket times 2 for an aneuploidy kit. And because you haven't got the nucleotides here, and it's also normal, you just put the kit name times 2 for, say, a subtelomeric MLPA kit. For an abnormal, where it's aneuploidy or partial gain or loss, again, it's still chromosome order, lowest to highest with X and Y first. RSA 21 times 3, or these are all correct. If it's male, RSA XY times 1, 13, 18 times 2, and because this is obviously times 3, that has to go in a separate order. It goes at the end because it's the last chromosome to look at. Or if it's female, it's much easier because these are all 2 copies. So you can put 13, X13, 18 all together, and 21 goes at the end. Don't forget, though, that if you've got something like chromosome 18, it will look even more complicated, and the easiest one is to put RSA 18 times 3, but you do need to say you've looked at the other chromosomes and which chromosomes you've tested in the kit in the text of the report. Or if it's female, RSA X13 times 2, 18 times 3, 21 times 2, but 21 cannot go here because you have to do chromosome order. And for a male, even more complicated because X and Y times 1, 13 times 2, 18 times 3, 21 times 2. And if you're doing the mosaics, then in this particular case, where we had times 3 here, it's times 2 to 3, or if you can quantify it, 0.6. So if you've actually got kits, if you've got partial loss or gain, because you haven't got a bracket, there's a space. You put the region, you put the gene, times 1, the region, the gene, and that's got 3 copies, which may represent a rearrangement, an unbalanced translocation. If you've got the coordinates, then you put the genome build, and again, there's a bracket, so there's no space, but there is a space there. That's the region, they're the coordinates, and there's 3 copies. If you don't know the genomic coordinates, then you put the kit, and for fusion genes, again, because there's a bracket, there's no space, it's BCR, colon, ABL1, and bracket negative or positive if it has got a fusion. This nomenclature of the colons will be adopted, and it will be published in Nature Genetics shortly. The whole genome array for a female, again, no bracket because you've got a no space because you've got a bracket. It's array X 1 to 22 times 2, and for a male, you separate the X and Y out because there's only 1 copy. For aneuploidy, you only describe the abnormal chromosomes. So for a triploidy, array X 1 to 22 times 3, or if it's XXY, you'd write it like this. And obviously, if it was XYY, that'd be times 1, and that'd be times 2. If you have an XXY, you would write it like this. If you had a near triploidy, so you effectively, obviously you don't know the chromosome number, but it would be 68, not 69, and you've got 3 copies of everything, but only 2 copies of 19 on the array, then you're only describing all the chromosomes except for 19 because that's normal, and you only describe the abnormals. And you don't give the genome build for normal results or aneuploidy such as trisomy 21. However, for structural aberrations, you do give the genome build. And there's two systems. There's a short system or there's a detailed system. Short description only includes the abnormal nucleotides. So you've got the array, you've got the genome build space, XQ25, they're the coordinates that are only in one copy. If you give a detailed description, you include the abnormal nucleotides as well as the bordering normal nucleotides. It does give more information, but it also makes it look more complicated. So the option is up to you. So in this particular case, these are normal. They say both say ×2. This is ×1, that times 2. The genome build must begin in the nomenclature if nucleotides are given. And the underscore is used to show the range involved in the loss or gain. And the platform information should be used in the description. I'm not going to spend time on polar bodies. Probably a few of you are actually dealing with polar bodies, but there is now a nomenclature, and we do have nomenclature for shallow sequencing, which is SSEQ, and you talk about chromatids, and you refer everything to chromatids. So it works the same way as arrays, except you talk about chromatids and you can use shallow sequencing. NGS, now this is interesting. Maybe it's because we're not so familiar with it. For a normal, you don't give ISCN, but you can use HGVS. The abnormals, structural rearrangements, you're describing the rearrangements identified by sequence-based assays, and it's a combination of ISCN-like and HGVS nomenclature. You only use the ISCN part if you can deduce the structural rearrangement. So in this particular case, you've got sequencing, genome build, it's a derivative chromosome, and then you've got the HGVS. However, the first line is the ISCN, second line is the HGVS. If you had a translocation, so you didn't have DIR2, it was just T2 to 11, you would have another line with NC, which would be describing what's happening on chromosome 11. This is a derivative, so there's only one line. And NC is the prefix used for chromosomes. And if you go to the HGVS website, you can find all the prefixes, but from a cytogenomic point of view, you're usually going to use NC. So to explain what this nomenclature all means, the chromosomes, I say, have a prefix NC. This is the reference sequence prefix. Use this to separate. Get these on the HGVS website. Use this to separate between the reference sequence file identifier, this is the file identifier, and the description of the variant. This is the reference sequence type, and this is linear genomics, so it's G. You actually have the nucleotides, which equate to the chromosome band. This is the type of variant compared to ref sequence, so a deletion insertion means deletions. And then this is the other allele. This is coming from chromosome 11. So you put these in square brackets, and then you again use the reference prefix, the reference sequence file, which is chromosome 11, it's still G, and then this is the breakpoints on chromosome 11. If you're not sure about the nucleotides, this is when you use the round brackets instead of the square brackets. So I talked about ISCNs now online. This is the website if you go into it. If you go onto the, if you buy the book, on the inside cover of the book, you'll have a unique file name which you have to use, and you sign in and you get access to the ISCN for a year. You click on this little button down here where it says Access ISCN Online. You will then get this page. You want to go to Table of Contents. You can go to the others, by all means do. But to look at what is actually in there, you will then, if you click on Table of Contents, get all the chapters, and you scroll down to see each chapter, and you just click on whichever one you want to look at. In some respects, it's not as easy to access as perhaps the book is, but in other ways, it's a very quick way of doing it when you haven't got access to the book. There's also now a forum. So if you click on this link up here where it says forum, you will get this page here. And there will be some of the questions are already in, we put some in, and you can start a new topic. And you can click on these to see the answers to question, and we asked Jean to reply to this one. Unfortunately, despite checking it, we found some errors. Now, the online version shows exactly where these errors are, and they're highlighted in red on the different pages where they have occurred. The errata will also be published in Cytogenetic Research, so there is a hard copy as well. And GenQA is going to offer a new pilot EQA, which will open in October, where you submit your ISCN answers on a pro forma. We will give you a sort of ideogram of the abnormality and ask you to submit the ISCN on a pro forma. You'll have 5 weeks to do it. There'll be assessors panel, which will include myself and others, and there'll be no performance monitoring because it is a pilot. So you can enroll now using www.genqa.org. As a pilot, it's much cheaper than a full EQA. We'll give a range of constitutional logical cases. You'll only have to do the ones that apply to you. There'll be a carrier type ideogram and fish images. And you have to explain, obviously, if you can't do all of them, to why you're doing a partial submission. This just makes it easier for us to mark. That's the reason we asked you to do it. So we're now on to the quiz. Melody?
Hello, yes.
Can you hear me?
So, yes, I can hear you. Can you hear me?
I'll start with the first one.
Yep.
Yeah, I can hear you. So, which is the correct ISCN description? A, is it B, is it C, or D? Now, Melody's going to put up a link for you to answer.
Yeah, so you should get a box pop up on your screen and you'll be able to select one of the answers. I'll give you a little while to think about it and press your button, and then we can close the poll and we can see how everybody voted. So this question, obviously we've got an extra chromosome 8 and an extra chromosome 9. So the question is, how do we write that? So the poll is open now. You should be able to see the 4 options. So please select the correct one. Okay, I'll just give you a few more seconds and then I shall close the poll. Okay, we've got about 3/4 of people have voted now. Here we go then.
So what answers have we got?
So can you see this screen, Ross? So the results are showing that 81% of the people who answered selected option C.
Well done.
So I'll just hide that, I think. Yeah, okay, so we can explain why option C is correct.
Okay, I'm having acute difficulty here. Oh, it's gone. Right. So there's a comma missing in A. There's 48 chromosomes for B, not 46, and there are extra spaces in D. That makes sense?
So, yep.
Correct. Right, next one. Which is the correct chromosomal order?
Okay, so we've got several different abnormalities here and we need to work out which order they go in. So here are your options. So a bit more to think about here. You've got numerical abnormalities, structural abnormalities, but we have 50 chromosome counts for each option, so you don't need to worry about that bit. Give you a few more seconds. So yeah, we can see the results more varied. We've got 53%, the majority are going for option B. Only 1% going for option D, and then 21% for A and 25% for C.
Okay, well, it's option B is correct. Sex chromosomes obviously go first. Autosomes go in numerical order, so the inversion 13 has to go first. Numerical go before structural only when it's the same chromosome. So you've got When it comes to identified abnormalities, the ring goes before the marker. I think that is it. So in this particular case, what have we got here? Oh, you've got a semicolon in, which shouldn't be there. In this case, the plus 21 must go after the inversion 13, and in this case, ring must go before marker.
Yeah, and
Next
that one's
one.
missing the plus 21.
Oh, yes, so it is.
Yeah.
I think that maybe. Yeah, it is.
Yeah.
Right, so of this particular one, where we've got a del 5, which of these is correct?
Okay, so there's 3 options here. Remember to look out for that punctuation again on this one. So I think this one's a bit more straightforward than the last one. A few more seconds. Okay, so we've got 3/4 of participants voting on here, and these are the results. So vast majority have voted for option A, with 15%
Yes.
for B and only 4% for C.
Again, it's no semicolon in deletions, duplications, or inversions within a chromosome in the short form. It's there for translocations and derivative chromosomes, but not for deletions, duplications, and inversion. And the 46XS line always goes last, even if it's the largest. And in fact, in this case, it isn't. So it wasn't even in chromosome. The largest clone wasn't put, cell line wasn't put first. So which of these is the correct karyotype for a balanced reciprocal translocation? Constitutional.
Okay, so you've got 4 options again on this one. Please select the correct answer. Okay, I'm going to close this one. So these are the results. So nearly 3/4 went for option C. The next most popular is 17% for option A, 9% for B, and just 1% for D. So we think it's option C, Ros.
It is C. A you cannot use because you don't use cell numbers for non-mosaic. B is incorrect because it should have a semicolon. C is correct because it's got the semicolon, and D is wrong because it's got a colon, not a semicolon. Again, it's the nomenclature. You've just got to be careful with your punctuation. Right, we covered this one earlier. So which is the correct karyotype for this complex rearrangement involving chromosomes 2, 3, 6, and 12?
Okay, so unfortunately we can't get the pictures onto the poll results. So I'll give you a little while just to think about this with all the information there, and then I'll pop up these options for you to select the correct answer from. Okay, so 78% have gone for option B, vast majority.
Correct. So as we mentioned earlier, you start with the lowest number. X and Y obviously go first, and the order of the other chromosomes depend on the movement of the segments. It's not a derivative, and it doesn't go in the numerical order. Last question. Oh, it's taking a while to move again. We will get to this last question. I'll move on to this. I think it's the easiest way of doing it.
Yep, I can see that now.
So this is a translocation detected between chromosomes 2 and 11 by whole genome sequencing. And we describe, because it's a translocation, both derivatives. So this will be looking at the chromosome 2, where it adds chromosome 11, and this is chromosome 11, where the reciprocal part of chromosome 2 is there. My question to you is this balanced or is it not?
Okay, so again, we can't fit all this information onto the poll, so we'll leave
You won't
this.
need to. Haven't you got the sequence part onto the next slide or not?
No, we've just got, is this balanced or unbalanced? So it'll be a
Oh,
yes or no. So I'll just give you a little moment to think about this one. This is probably the most tricky question. We saved that for the last one. Okay, we've got lots of votes.
Is it a 50-50 split?
It is nearly, yeah, it's a tricky one. So we've got 56% saying yes, it was balanced, and the other 44% saying no, it was unbalanced. So which is it, Roz? Is it frozen
it's
again?
Unbalanced. There's a 5 base pair deletion. If you look at it's getting familiar with it. If you look at the coordinates on chromosome 11, they're out by 5. They're the nucleotide numbers, so that's why it's unbalanced. It does take a lot of getting used to, but hopefully this will help you to understand it a bit more. Those who are using it routinely will be quite familiar with this. So just to give acknowledgements, there'll be a bit more information in a minute. The ISCN Standing Committee, because I'm just part of this group, although I'm giving this talk, there are plenty others that could have easily have done it. Cargo, who've obviously done the printing of the book and provided now an online interface plus a forum for us to discuss issues. GenQA staff for obviously facilitating this and helping and giving feedback, plus yourselves who are listening, because without you, we don't get feedback about what needs to be improved and we don't adjust the nomenclature. So keep sending that feedback because it's helpful. So all I can do is say thank you. But before we go, do you have any questions or questions for us which we can answer, hopefully?
Yeah, we've got a few coming through on the question box on the website. So if we start with a couple about marker chromosomes, so we'll link these together. So there's one question which is what is meant by marker, and the other one is if you know the identity of the marker, how come it can still be classed as a marker, and could it be a derived chromosome?
Right, I'm going to have to repeat that second one again because they're right on both counts. The first one, marker, is an unidentified chromosome material that you cannot work out what it is, and so you call it a marker. Sometimes people call it a supernumerary, remember it is
Supernumerary
an extra
marker chromosome.
supernumerary marker, yeah. But we just tend to refer to it as a marker chromosome in ISCN. Now, what was the next part of the question?
So if you know the identity of the marker, how can it still be classed as a marker, or could it be a derived chromosome?
Well, you see, this is where this is where the G banding comes a problem, because if you've got it, say, of G banding, you've got, say, 47XX plus more, and then you do FISH or you do array and you identify it. You only in hindsight can change the G banded to what it is if you can see it on G banding. And if it's small, you won't be able to say, oh, yes, I can see in hindsight it was chromosome 8 after all. So you have to have it as its full chromosome identification in the FISH or the array result. And yes, it can be a derivative chromosome. It can actually be sometimes an unbalanced segregant that you haven't identified previously, or it could be de novo, of course. So yes, it can be all those things.
Okay, thank you. Right, we've got an interesting one here. So are you planning to develop ISCN nomenclature for NIPT? e.g. maternal versus fetal abnormality?
No, we are not going to do it for NIPT because that's a screening test. And you can't, if you start doing ISCN for a screening test, then you're implying that that's a diagnostic test, and it's not. We can do it for NIPD, but not for, and then you would probably use the array nomenclature, but we can't do it for NIPT.
Okay. So I think we alluded to this one in the talk. If you know a chromosome is abnormal, but you can't identify what the abnormality is exactly, how do you write the ISCN?
Well, it depends what you actually can see. Can you identify any of it? No. Marker. Can you identify part of it? Describe the first part of it as part of a derivative and put query for the second part. Does that make sense? Or add. You can use add.
Yeah.
Add, people don't like, some people like using add, some people don't. Add means there's additional material on the end of a chromosome. It doesn't mean that it's bigger in size, it can be smaller. But it is quite useful when you're unsure whether it's actually a derivative chromosome or another chromosome is involved, or it's actually the same chromosome material. It's, you know, say it's an add 8, you don't know if it's coming from part of 8 or it's actually another chromosome altogether that's on the top of 8. You know, it could be a duplication, for instance, of chromosome 8.
Yeah, and like you said in the talk, you put the question mark in. If you can identify some of it, you put the question mark before the bit that you're not sure about. I think there's just a few comments. Yeah, so she wasn't referring to an add, was a structural interstitial abnormality.
You can't do it for a structural interstitial for add, because it only does it for the end of a chromosome. It doesn't do it for an interstitial. It's a terminal band.
Yeah, so you could maybe describe the chromosome as a derivative chromosome with question marks at the breakpoints, yes. I guess, where the interstitial segment is that you don't know what it is.
That's right. Yeah. There's some examples on page 64 of the ISCN book that might help. Somewhere they've got one here with a DIR7, T27, and then they've got query. I've got insertion 7 semicolon query. But there are some examples in the ISCN book. If they've got specific examples, stick them on the forum, and we'll answer.
Yeah, so we'll just do a couple more.
Description of what you're seeing, because it's very difficult for us to do, you know, make these comments when we can't see what you're referring to.
Yeah, so we have overrun, but I think we'll just answer one more question, a specific question, and then some of the other ones are general ones, which we can cover in a minute. So it's a sequence-based one. Should you use the term balanced or unbalanced for indels?
Well, you'll know that from the nucleotides, so then you can say balanced or unbalanced.
Yep. Okay, and if we, so some of the other, was just one more here, for the pilot ISCN, will that include RSA and NGS? So I can answer that one.
You can answer that.
Yeah, so we're just working out what our software will be able to display. We definitely should be able to include some RSA examples. I think the NGS we might leave out for this pilot. That might be covered elsewhere anyway. But we want to focus really on just giving a range of results for participants, see which questions they like to answer. And what we'll look at doing in the future is splitting up the EQA into more discipline-specific areas. So like with these talks, a constitutional aspect of the EQA and acquired, we might separate it by technique. But for the pilot, we just want to give a range of questions and see how it goes. Okay, so any other questions that we haven't had time to answer? we can have a look individually and either respond to you by email, or we can publish something on the website. If we just move on to the next slide, Ros, because I think this will cover a couple of the other questions, which were more general. Or is it frozen again? Well, anyways, what we were going to say is...
I'll do it. Sorry, I forgot.
So as a registered participant, you will have priority access to this webinar. It has been recorded. You'll be able to view it for a month exclusively. If you had any technical issues or if you couldn't log on from the beginning, that will be available to you on YouTube. Following this, it will be available on the platform. Following this, it will be on YouTube, and anybody who hasn't registered can subscribe to it. So if you feel that this would be useful for one of your colleagues to view, they'll be able to access it for free on YouTube in about a month. You'll be given a participation certificate automatically by email by the system. It won't be sent to you immediately, but hopefully in the next couple of days you'll receive your certificate through. We will be doing another Focus On session in September, which will be on lymphoma. But of particular interest to these registrants, we will also be offering an ISCN focus on the acquired abnormalities in October. If you need any further information from us, please see our website or email us directly. Hopefully we'll be able to answer any of your questions. And if it is something very ISCN specific, as Ros has mentioned, you can use the forum for that as well. So I think that's everything for today. Anything to add, Ros?
No, thank you, Margie. I think you've covered everything. And thank you all for participating. It's been wonderful to have such a good response.
Thank you.
Have a good rest of the day.
Yep. Okay. Bye.
Bye.